His company for 18 years was incurring losses. But he believed in it, right? He persisted with it. The other characteristic of people who succeed are people who were for persistence, who have tenacity, who never give up, right? And this all things I'm sure you've listened to, you've heard, you've listened to in all your lectures, but it really comes down to those very simple things. Having a vision, right? Having a vision to do something, having a dream to do something, and then working towards it. I mean, creativity is God gifted in a lot of ways. You can't, I mean, you're born with it. I feel, but that's my belief. It's 2:15, brother. Okay, so you guys, when you want success in your life in whatever you do, make sure you remember that. This might sound like this guy. Lots of people say these things, but actually, what happens is when you've lived life enough longer than you guys, you realize that these things are the ones that matter. Okay, so please think about that. When you want to succeed, think of dream the dream. Don't give up your dream, right? So day after tomorrow, I want to hear your dream. Okay, tomorrow I'm going to hearing your 5-year plan, but day after tomorrow I want to hear what you dream to be in life. Okay, everybody, you can make it up your 48 hours, less than 48 hours. Okay, but tell me what your dream is. Okay, dream, work towards it. Don't give up, right? And believe in yourself. If you believe in yourself and your abilities, and some point you realize this is the end of my ability, and that's okay. Living a good life is good. That's fine. You need to ensure that you believe you try your best, right? I'm sure your parents say try your best, right? When you go to do an exam, try your best. You do your life, try your best. That's okay, right? But some things will materialize, not everything. Your dream may not come true, but the dream, something towards the dream will come true. You'll be satisfied. When you're when you're dying at the end of your life, you need to be satisfied with yourself. We were just talking, right? I mean, it's everybody can make money. Everybody can find their bread. Every single animal on this planet, till he lives, finds his bread, knows how to live. We are all animals at the end of the day. We will always find ways to live. We will always find to make money. That shouldn't be the goal. Goal should be what you really plan to achieve. Do something that is meaningful. Otherwise, there's no difference between you and the animal who's just living for his bread, right? Okay, all right, let's go back to chromatin. We're talking chromatin now, okay? What does chromatin mean? You guys know chromatin? What does chromatin mean? Somebody gets up. Stand up, one of you. You know the procedure, right? No? Nobody knows chromatin? DNA plus histone is chromatin. Okay? Anybody else? Chromatin. Chromatin is my favorite word. Why do we have this slide and why am I talking about chromatin? What does the slide say? I'm comparing a eukaryotic gene regulation or transcription to a prokaryotic transcription for a reason. What does the slide say? Anybody knows how to read English? No? Half the people went to sleep, I guess, after eating, right? The gene transcription was going crazy. Okay, this says prokaryotic gene ground state is on, and the eukaryotic gene transcription ground state is off. What does ground state mean? Ground state is a state in which, right, the genome normally lives. Prokaryotes, normal ground state is on. That means they keep on transcribing. Whereas eukaryotic system, everything is off and it goes on to become on. For all that we studied in the previous 2 hours, right, was how does a gene transcription goes from off to on, right? Okay, that's chromatin-based regulation. We studied the right side of things. You guys know that, right? On the bottom right, you can see that. Good. Chromatin maintains the repressive state, the ground state of. Prokaryotes don't have chromatin, right? Eukaryotes do. I mean, I'm making general statements. Some of it might not be true for some things, but whatever, at least chromatin maintains the restrictive ground state for transcription. Because the eukaryotic cells, as they evolved, they decided we don't want to be transcribing all the time and killing our energy because we have lots of genes. How many genes prokaryotes have? Few, very few. How many genes bacteria have? How many genes COVID-19 virus had? You guys lived through a great calamity in our lives. Once in a lifetime opportunity, once in a blue moon, once in several decades, right? Once in 100 years phenomena. You were lucky enough to live through COVID. I'm not kidding. Right? आपने एक महामारी देखी अपनी जिंदगी में. Seriously, this is not funny, guys. This is sad stuff. You saw a disease killing lots and lots of people within hours, right? I'm sure many of you guys had families affected by it, right? How many genes does COVID-19 virus have? Do you guys know? How big was the genome? Where is curiosity, guys? Curiosity. Virus that killed a lot of people around you. You didn't care about it? The virus that put you in the home for months. You don't care about how many genes this stupid trans virus has? The virus that we cannot cure? A virus that whole world, put everybody in the world on their knees? Know how many genes COVID-19 has? 30. How many genes a human has? 23,000. Look at the complexity. Look at the complexity. How much what was the size of the COVID-19 virus? 30 megabases, 30 megabases, up something approximately. I'm not saying, don't quote me on exact numbers. But I'm just trying to tell you the complexity difference between a virus and a human. And we cannot cure a thing there's only 30 genes. What are we going to cure cancer with 23,000 genes? The challenge, guys, in front of you. The God gave you the challenge to solve. The God gave you the challenge to solve. Okay? I just want to ensure that you guys understand the complexity of human genes because it's important. It's important for what we're doing, right? So chromatin, we didn't talk about it before this, right? We talked about transcription. We talked about transcription factors. We talked about regulatory elements. Does anybody remember? Everybody was hungry. What did I say was the most important thing? Answers. Okay, good. People were not sleeping, and the ones who were sleeping are still sleeping in their rooms. Boom. What is this? Why did I put that out there? Nucleus, eukaryotes, prokaryotes don't have nuclei, do they? Nucleus is a great regulator of things, right? Why is that a great regulator? What's the role of nucleus in a cell? One person, stand up and give me your answer. Nucleus controls metabolic activities of the cell. That's not really why. That's not really why. In my introduction, when everybody was sitting, all the dignitaries were sitting, I said what? I said I have a hypermetabolic syndrome, mitochondria syndrome. Why did I say that? Because mitochondria is where lots of energy is produced, isn't it? Okay, just kidding. But nucleus is not, because nucleus just Okay, why nucleus? Anybody else? Yeah, please sit down. Yeah. Why is a nucleus a regulator? Wonderful. Thank you. Nucleus is its own phenomena inside. It allows only certain things to come in, right? Your DNA is inside the nucleus, isn't it? Right? So nucleus itself is a regulator. The nuclear transport is a big regulator of things. All these nuclei have nuclear pores, right? Only certain things can go in with a passive mechanism, less than 45 kilodalton worth of things. Okay? You think I have less things to remember? Okay. And this is what we did yesterday. What is this? Chromatin. But what is that? Huh? Noodles in a bowl. What is it? Interface nuclei. Thank you. You were paying attention yesterday. So events of the transcription are taking place inside the nucleus. Of course, we know all of that. And what's inside the nucleus? Beads in a stringer, right? All right. So basically, you have chromatin around which DNA is wrapped, which we all know. We've talked about this. Nucleosomes acting as a roadblock for transcription. That's why it maintains the ground state, right? That's really what we're talking about. So the access to the promoter or the enhancer or whatever is not available in certain scenarios by chromatin, because there is a chromatin compaction. There is a nucleus, there is nucleus. This is a 10 nanometer fiber, right? Okay. So in the top case, you have the excess of transcription factors for DNA. In the bottom case, you don't, because there is nucleosomes next to each other. And that's why chromatin accessibility is critical. That's the problem we're going to solve today. That's the problem we're going to solve. The problem to solve is how to make transcription factors and DNA binding factors accessible to the DNA. DNA is not accessible because it's wrapped around a big protein, right? That's the problem we're solving, because that is a critical regulator of transcription by very simple means of not allowing your DNA transcription factors, your RNA polymerase to bind and move through, right? That's a big, big, big problem. That's what we study today. That's what we'll study day after tomorrow when we talk about nucleosome remodeling with 3D chromatin structure, da da da. Right? You guys have seen this? So the problem we're going to solve in transcription that regulates is through accessibility of the DNA, right? Very simple. Accessibility of the DNA is a big regulator of things. To just solve that very simple problem, cells spend lots and lots and lots of energy in transcription, but that's the way to regulate genes, unfortunately. So it's not a very efficient way of doing it, but that's how we are built. Maybe that's why we live for a certain number of years. I don't live for 1,000 years. Okay. Anyways, that's not true. I just said that. Okay. But okay. So here is a heterochromatin. What does heterochromatin mean? I'm sure you guys have read this. Huh? Highly condensed part of the nucleus. This is an electron microscope picture of a nuclei, right? So real stuff. This is how our cells look. If you just put it under the electron microscope and you look, don't stain for anything. You're just looking. You're looking, does it look like white? Does it look like black? Right? I can put some fair and lovely and I can make this white, and then it will become euchromatin, right? Heterochromatin, euchromatin. Euchromatin is the white piece, heterochromatin is the black piece. All right. Densely packed is heterochromatin. Great. Heterochromatin. Boom. We know the answer. Kaun banega crorepati. Loosely packed as? Very good. Chromatin fibers. We'll talk all about this again on day after tomorrow, but I want you guys to appreciate. This is again an electron microscope picture, very famous. Wikipedia. You can look up all of this. The top one is condensed. The bottom one is less condensed. Your beads in a string, your necklace, right? That's a 10 nanometer fiber, the bottom one. Okay, right? You guys know this stuff, right? I'm just repeating it. Most of you, everybody, tell me, raise your hand if you don't know this. People are like, oh, I know this. Okay. This is 30 nanometer fiber, lots of this packed with each other. You're not allowing things to go through. That's my restrictive state. Okay? That's not allowing anything to happen. And then they like to make these beautiful structures, 300 nanometer fibers of sorts. Not entirely true all the time, but yes. Okay? So that's the layers of chromatin structure. That's how it looks. It says possible level of complexity. Makes it easy for you to comprehend, but that's not always true, right? Okay, good. So we looked at the DNA in a pipe, nucleosomes, chromatin fibers. 300 nanometer, 700 nanometer chromosome. I don't know if that is all true, but whatever, right? For your simplicity, believe it all to be true. Okay, cool. Anybody has doubts here? You can imagine this in your head in an interphase nucleus. This is how 2 meters of DNA is packaged into nanometer of space, right? What a beautiful cell, huh? What beautiful nature. How did they think of this? Right? This stuff boggles my mind. Wow. Every single time there is a metaphase, they're going to make that from interphase, right? Every single time the DNA will be packaged, it will go through the rounds of doing this and then make your chromosome will be segregated, will go back, then will go back to whatever state it was in, back to that. That's called heritability. That's epigenetics for you, the genetics part of epi, right? We got this? Every single time. And if it does not happen, then what happens? What happens if it doesn't do that every single time? Stand up and answer the question. Loudly. Dysregulation will happen. Of course, good answer. What kind of dysregulation? Let's look at Google, AI this stuff, right? Can you figure it out? What will happen? Come on, guess. What will happen? If you are a cell and something goes wrong, not according to your plan, what happens? USA, you're going from here to Amarkantak and you want to reach there in 3 hours and there is a huge rain and the road is blocked and you can't reach there. What happens in your brain? What happens? I can't hear you. Stress. Are you angry? Does the cell get angry? What will happen? Imagine what will happen. You're a scientist. Imagine we don't know. And in fact, we don't know actually. Humans don't know what happens. What happens if the 3D structure is not solved every single time? What happens? Now I'm giving you the challenge. Think of a problem. What will happen? Write a research project on it. Build a hypothesis. Are you guys taught the method of building hypothesis? No? Seriously? You are given a problem. You need to come up with a solution, right? What do? How many of you watch thrillers? Right? Everybody loves thrillers, isn't it? What's your most favorite thriller? Huh? Detective movies. What was that Ajay Devgan movie? Drishyam. What was the detective thing in it? Did you guys figure it out? Like when you were watching the movie? Was it very interesting, right? You had a hypothesis. Did you have a hypothesis when you were watching the movie? Oh, maybe this happened. Oh, maybe he's the killer. Or maybe she is not the killer. Well, you knew who was the killer in that one. But in other movies, right, you don't know who's the killer. right, or whatever, but you have a hypothesis. Your hypothesis is based on the evidence you see in front of you that the director is showing you, right? So you say, oh, I'm sure his brother is the killer. I'm sorry, think of a thriller, good thriller movie. I don't know, I'm bad at this. What's your most favorite thriller movie? One, any of you? It's okay, we're having fun. Huh? Shawshank Redemption. Okay. What else? Sorry, I know Shawshank Redemption, but anywhere where there was a killer, the killer was the next door guy they never thought of, right? This happens all the time, right? It was the mother, is the wife, is the whatever, right? They take their revenge, but you are always thinking that, okay, maybe this person is the killer. So you have a hypothesis. Same thing with experiments, right? I'm sure you know how to. PhD students, you know how to do evidence, right? Look at evidence, find build hypothesis. You don't? You do? Your teachers don't teach you this? I'm sorry, I'm saying this. Well, I'm sure that they teach you, right? You have to think of the problem, the evidence is there. You think of, okay, then you connect the dots and say that's your hypothesis. Your hypothesis may be true or may not be true. Sometimes you think, oh, he is the killer, but actually he doesn't turn out to be. There's somebody else is the killer. So you make a hypothesis based on the evidence. Make a hypothesis. Yes.
Since the two sister chromatids they separate from the centromere, so if this level of complexity is not reached, then there will be unequal division of the DNA into the two daughter cells, which might lead to some mutation or some defect.
Great. He's the only guy who reads. Right? This is how cancers happen, right? How do cancers happen? Have you guys ever wondered how do cancers happen? Mutations. Everybody's out of mutations. How is the mutations happen in the cell? And one person stand up and give your answer. It seems like everybody knows this. Somebody who I never heard. Who knows this? Raise your hand. I'm going to pick. What is a mutation? Raise your hand. Not the regular. Okay, you. I have never heard you, man. Go on. Any kind of deletions or insertions in the, or the change in DNA base pair sequence. Great, that's mutation. Very good. How do those happen? Carcinogens. Many great. Many carcinogens make mutations, right? Good job. UV light makes mutations, pyrimidine dimers, things happen. But then how does that get passed on? Why do we not correct it? Why does the cell not correct it? Or does it? Does it correct it? That's called what? DNA repair. DNA repair, right? We got a chromatin damage, right? So there's a DNA damage that you guys talked about. DNA damage gets repaired by the machinery. So we've got a chromatin damage problem. I don't think people understand how chromatin damages are repaired. Okay? Great problem to solve. Okay? My budding scientists, go on, figure out how we could do this. All right, that was that. That was just to make you guys think a little bit. What is a nucleosome? DNA plus histones is a nucleosome. Why am I showing you this? Nucleosome is the building block of chromatin, right? Our beads on the thing is a beads in a string, everybody knows, right? Okay, nucleosome, my favorite molecule. I put my passwords as nucleosome. Don't try to hack my account. Okay. Okay, a nucleosome is what? 1.67 tons of DNA wrapped around 8 histone octamers. I'm sure you've read your books, right? 200 base pair, actually 171 base pairs. Okay? And then there is H2A, H2A, H3, H4, two of those, each of those That's called histone octamer. Why is it called octamer? 8 proteins. Great job, guys. 8 histone core. That's a nucleosome. What's that thing on the side? Not all nucleosomes have linker DNA, linker histones, by the way. Only few of them contain that. So we made that. That's just a cartoon. That's not entirely true. But some of them do. Histone H1 is used for chromatin compaction a lot of times. What's this? Why am I showing you a bunch of alpha helices? Huh? Histones. This is nucleosome, right? This is histone octamer. You see 2 copies of each. Do you? Okay, good job. It's a left-handed supercoaltans normally that are around this. Again, that doesn't matter. There's a positive charge and a negative charge. That's important. That's really important. What is positive? Histones. Stones are positives. Why? Okay. What is arginine? It's an amino acid. Wonderful. Why is it positive? Basic amino acids. Okay. Good. What's negative? Why is DNA negative? Ah, you guys know all this stuff. Great. That's crystal structure of nucleosome. What's the crystal structure? What's the crystal structure, guys? Okay, keywords. Don't throw keywords at me. What is the crystal structure? Okay. All right. Homework. Go read about crystal structures, please. I'm not going to tell you about how to solve a crystal structure. Neither am I an expert in it. But I want you guys to look at crystal structures, right? Okay? Please, homework means go out. Be curious. Go figure it out. Okay? These days, people do something called cryo-EM. Have you heard of cryo-EM? What's cryo-EM? You nodded your head. What is cryo-EM? What is cryo in it? Okay. Really cold temperatures is cryo. Okay. That's a histone. Very simple. You see the histone structure? That's how it looks. Right? That's why I was showing you all those bunch of alpha helices every single time. Right? That's a histone. There's a lysine and an arginine. We talked about this. They are rich in lysine and arginine, so they are positively charged. Histones are found in eukaryotic cell nuclei. You know all of that. This is how histones look. H2A, H2B, H3, H4. Histones are the most abundant protein in your cell. How many base pairs we have in the DNA? In a eukaryotic genome, let me put it that. How many base pairs? 3.3 billion. How many bases per turn of nucleosome? What? 171, right? How many nucleosomes should be there? 3.3 billion. Divide that by 171, all the math geniuses here. Right? Think of how many different nucleosomes are there. Nucleosomes are very abundant. Well, not every 200 base pair is a nucleosome, so yeah, we can't really divide them. But you get what I'm trying to get you to think. Imagine this. Just imagine, right? You've got lots of nucleosomes around which DNA is wrapped, and each nucleosome is 8 histones, right? So how many histones are there? Oh my goodness. Cells make a lot of histones. Easiest proteins to detect on a Western blot. What's a Western blot? What? Protein separation, Western blot. Why is not Eastern blot? I'm just kidding. I don't know the answer to that. Western blot. You guys have heard something called Northern blot? That is used for? So everybody's read the books. I like this. Good. What about Southern blot? Why not Eastern blot? Ah, great. Thank you. See, I learn everything things from you guys. Great. My basic principle in life is to learn something from a person in front of me. So thank you for teaching me that. Okay, I didn't know that. Okay, that's why I want you to call me Kunal. You you can't teach me something if you keep calling me sir. Then I'll have to call you guys sirs and ma'ams, right? But if you come at my level and you call me by my name, I feel comfortable learning from you. You feel comfortable giving me the knowledge. That's why it's important. Okay, get it? Why it's important to be at the same level. You respect me because I'm older to you, which is a great thing our Indian culture teaches us. But you can call me by Kunal. By calling me sir and a hierarchy between you and me, I don't get to learn from you. And I would like to. Right? My basic principle is to learn from people. That's how I get my knowledge. And I'm in the business of learning, aren't we all? Right? We are all in the business of learning. Okay, that's histones, right? You got this? 3 alpha helices, each one of them. Not each, yeah, more or less. H2B is different. H3 is different. Okay. This is where all the magic happens. Histone tales. Why we are doing this class. Why I'm spending 4 hours with you guys. Why I'm spending my life with my research. I do a lot of histone modification things in my lab. What does it show you guys? What is in the slide? What? What does it show you? Okay, let's get 5 answers from you. Each one of you. I want 5 people to stand up and tell me what you see. One at a time. Modification of histones. All right. Next person. Acetylation and methylation of different amino acids. You mean you're not seeing the phosphorylation? Okay, next one. Anybody else? I said 5. Come on, guys, in this class I can have 5 people stand up. There are only 3 amino acids that are showing methylation and acetylation and phosphorylation. What are those 3? Lysine and arginine and threonine. What's threonine? Okay. 3 people. I need 2 more. Show me your guts. We're talking about risk taking. You can stand up and tell me what you see on the screen. You, go on. Methylation? Methylation is on different amino acids. That's true, arginine and lysine. And the one behind you, what do you see on the screen? Okay, anybody else sees anything different from what everybody said? Yeah. Good job. What else? I'm looking for a specific answer that's hidden. I'm seeing the detectives here. A transcription process. This slide does not show you transcription process, my friend, but I admire your answer. Huh? That's not on the slide. It will come though. Thank you. There is no ubiquitin on this. He does not see ubiquitin. That's an observation, okay? All right. I'm not looking for any of that. I'm looking for something else. Particular number of amino acids get methylated. That's a good observation. Thank you, but I'm not looking for that either. Anybody else? Not all bases are getting modified. Great.
So serine can show the phosphorylation and the lysine can show acetylation and methylation is the arginine.
Great. So you guys are telling me that specific amino acids get modified in specific manner. That's what you told me, multiple of you. I'm still not looking for that. I'm looking for something else. Looking for these keen observers between you.
Methylation and acetylation occur in single amino acid, but phosphorylation occurs only in the
Okay. Great. Anybody else? The detectives. Thank you. Good observation. Thank you. I'm not looking for that either. But you guys are making good observations. I like it. I like it. I like it when my class is engaged, looking at things and telling me great things. Yes, please.
So serine is a polar amino acid, that's why phosphorylation can happen and methylation can happen in lysine and arginine which are positive.
Good. Very good. Very nice observation. Lovely. That's still not what I'm looking for. There is something more. Okay, keep going. Amino acid residues with A do not get modified. Good observation. There are some that do not get modified on this. Wherever acetylation happens, simultaneous methylation happens. That's a great observation. I'm still not looking for that. I'm looking for more things. All fantastic observations, guys. Amazing. Amazing what you're telling me. I love this. You know why? Because I'm going to tell you about something called histone code. This is detective stuff. This is fun. This is frenzy. This is what got me interested into doing what I do. This was my first year of my PhD. I love the coursework because this is so brilliant. Simple proteins. Okay, I want you guys to go home today. Okay? Well, I will have to tell you this later, but go home and read about histone code. Okay? It's a paper by somebody called David Alice, and a genius guy who just died like last year, I think. Brilliant guy with Rockefeller University. Histone code. I want you guys to read the paper. It's brilliant. The guy was just genius. Okay? Do you guys see a pattern? Pattern on the sequence. Do you? See a lot of AR? There's a song, right? Likhna haathon mein A plus R, something like that. Lucky Ali. Okay, I want you guys to go home and think about whether there's a pattern in histone sequences. There are patterns that might not be as obvious to you, but there's a pattern. So every few bases are same or similar. Okay? So therefore they get this pattern. So you see there's a periodic way they get methylated or acetylated or something. Okay? So ARKS, this sequence happens a lot of similar. You see ARKS? Couple of them at least on this tail. This is histone H3 tail. Histone H3 tail is most heavily modified histone tail with covalent modifications, methylation, acetylation, phosphorylation, butyrylation, succinylation, propionylation, da. There are 150 modifications known that occur. Okay? How many? 150 is an old slide. It does not show you ubiquitination. Somebody said that. Great observation, because ubiquitination happens inside the histone octamer. Okay? It happens on lysine 119. There are not 119 amino acids here. See that? It happens inside, somewhere there. Good. Good observations. I love that you guys participated. I love that. That's what I want you guys to do. Okay? Speak up, because there is no wrong answers. Okay, how do these modifications take place? We'll learn that. What are the consequences of histone modifications? That's even more profound. That's how gene transcription regulation happens beyond everything else I've told you so far. Okay, this is a slide I use. This is one of the slides I have. The previous one was Arpan's slide. Okay, that's an H3, H4, H2A, H2B tails that are shown you here. Again, same thing. You see that, right? It also shows you methylation on DNA, by the way. It also shows you some other things there are which are called non-coding RNAs. So the slide summarizes a lot of epigenetic modifications. So you have lots of DNA modifications. There are 150 different kinds of modifications happen. Now think about this. Histone octamer has 2 of each. These amino acids get modified, right? My mathematicians, how many such probabilities are there of happening? What kind of states? So chromatin nucleosome can be modified in many, many ways, right? And think of how many different probabilities one can have. Wouldn't be cool? You have 2 of each. So you have 2 tails. One could be modified differently. One could be modified differently. You have so many amino acids that get modified. The specific combination they occur in. If you have an acetylation, you can't have methylation on the same. If you have a serine 10 phosphorylation, usually K9 methylation does not happen. That's just shown here. So cell has built this beautiful logic inside with beautiful sequences next to each other, and it knows where to put what mark at what time in which gene, and that's how it regulates gene transcription. Okay, this is an old slide, but here I want you to appreciate that there are different kinds of modifications that result in different outcomes. Okay, so methylcytosine, DNA methylation that we studied yesterday, we'll study more tomorrow. A lot of that is a gene repression mechanism normally. Acetylated lysines are always associated with positive transcriptional activation. Phosphorylated, that's take with a grain of salt. Methylated arginine, that can have both. This is an old slide. There's more data that shows it can do both. Methylated lysines make the magic happen. It depends on where they are happening. If it happens on H3K4, histone H3 lysine 4 residue, they're always active marks. They want this, they mark the gene to be active. Boom, boom, boom. Okay? And the ubiquitinated ones can do both. They're inside. But histone H3, sorry, let's go back to the methylation. Methylation or lysine 9 would be associated with repression. K4 activation, K9 repression. They're very different. They look very similar, don't they? Let's go back to this. That's the K4. That's the K4 methylation. Okay? That's K9 methylation. See how different they are? ARKS, ARTK, whatever, right? Close to each other. They look very similar. K9, K4. So if you put in the 4 one, the cell is always associated with positive transcription. In fact, all the enhancers are made by H3K4 monomethylation. Mono means 1. The funny thing is, I didn't tell you this, right? Let's come to that in a second. Let's see if I have that here, because before I go. Oh, we'll come to methylation actually. I'm sorry, I'm hurrying ahead of myself. Okay. We'll come to this then later. Let me go back to make it simpler for you. I'll come back to the slides, okay, afterwards. Okay, general themes of DNA, any type of chromatin modification. So we asked the question, how are these modifications made where they are? Take a guess. I want to put a mark on. What will I do? I'm a cell. Use your intelligence. I'm a cell. What would I do? I want to methylate 4. Take a guess. Simple stuff. What kind of enzyme will I use? Methyl transferase. Good job. That's what all these MTs mean. Methyl transferase. Histone, lysine, methyl transferase. I want to remove it. What will I call it?